to keep their mic on mute. Let the speaker
Yes.
complete a session and we will have a separate question answer session at last. So I,
Okay.
Rahul Sankla, on behalf of CCS National Institute of Agriculture Marketing, Jaipur, welcome you all in KisanMitr webinar series, Innovative Lab to Land Technologies for Indian Agriculture. Today we have a session on this topic. So first of all, I just introduced to CCS about CCS NIAM. CCS National Institute of Agriculture Marketing, Jaipur is an autonomous organization established under Ministry of Agriculture and Farmers Welfare, Government of India, to cater various agriculture related aspects such as training, research, consultancy, education in agriculture marketing. CCS NIAM is also recognized as globally as the center of excellence in Southeast Asia. KisanMitr, Friends of Farmers, is an initiative by the Principal Scientific Advisor Officer in partnership with CCS National Institute of Agriculture Marketing, Jaipur. The objective of the initiative is to bring technologies to farmers and reverse migrants who have moved rural due to the impact of COVID-19. Under this initiative, more than 1,000 technologies have been identified which are supported by ICAR institutions, NABARD, Ministry of Rural Development, ISRO, and Gates Foundation. So on the supply side of the technology, there are innovators, entrepreneurs, research in academic and research institute talking about their technologies, and the webinar participant being on the demand side, who are typically agribusiness, government functionaries, agribusiness companies, and other agribusiness stakeholders who are keen in agriculture technologies. Moving further, I would like to introduce our today's eminent speaker, Mr. Jagan B. Bhale Rao. So, he is a scientist. Just a minute. Yeah, sorry. So, moving further, I would like to introduce our eminent speaker, Mr. Jagan B. Bhale Rao. He is a scientist and head Department of Biotechnology in Ellora Natural Seeds, Aurangabad. And he is also working in specially molecular biology lab as well as quality control lab for testing of hybrid purity, parental purity, seeds, germination of different vegetable crops, oil crops, pulses crops, etc. So now I would like to invite our eminent speaker Jagan B. Balerao to take a session on innovative lab to land technology for Indian agriculture. Over to you, sir. Thank you. Mr. Jagan B. Balerao, sir. Over to you. Voice is not coming, sir. I request you please unmute your mic, sir. Jagan, sir, your mic is muted.
Hello?
Yes, sir. Audible,
Hello?
sir. Audible, sir. Audible,
Yes.
sir. Please go ahead, sir.
Yes. Good morning, all of you. Myself, Jagan B. Bhalerao, senior scientist and head Department of Biotechnology in Ellora Natural Seeds, Aurangabad. I have currently working, our company currently working in total specially vegetable crops, such like chili, tomato, brinjals, onion, focusing on specially vegetable crops. I am working biotechnology lab, a lot of experience, biotechnology lab, molecular lab, as well as quality control lab, tissue culture lab, especially banana tissue culture, pomegranate tissue culture, and other are molecular marker technology lab, such like totally hybrid purity testing, parental purity testing of different crops, vegetables, pulses, oil seeds, etc. I have shared my PPT. Hello,
Yes, sir. Screen is
hello.
yes, sir. Now presentation is visible, sir. Go ahead, sir. Good
Yes,
to go,
yes.
sir.
Uh, okay. Welcome to all of you. My webinar topic: Innovative Lab to Land Technologies for Indian Agricultures. Firstly, importance of seed testing. Seed testing has been developed to aid agriculture to avoid of the hazardous crop production by furnishing the needed information about different quality attributes, purity, moisture, germination, vigor, and health. Quality control of seed depend on the different seed testing protocol which determine the genome of cultivar. Testing of seed evaluate the planting value at the authentically of the certified lot. Seed testing is required to assess the seed quality attributes of the seed lot which have to be offered for sale. These quality attributes are seed, moisture content, germination, vigor, physical and genetic purity, freedom from seed-borne disease and insect infection. In India, seed testing is done mainly for moisture, germination, and physical purity of seed are generally determined. Seed testing is basically specially on in laboratory for purity testing, moisture testing, germination testing, vigor and health. What is the health of seed testing? Such like chili, tomato, brinjal, in vegetable crops as well as oil seed, uh, sunflower, pulses like green gram, black gram, etc. Standard seed testing procedure for the evaluate of seed were developed by ISTA, International Seed Testing Laboratory, is the developed procedure evaluation of seed testing. It is obligatory on the part of seed analyst to follow rules prescribed by ISTA in 1985 if the seed is moving to the international trade. The seed testing procedure which are described below are based mostly on the international rule because most of our rule are based on their ISTA laboratory. As per ISTA 1996, economic yield of a crop depend on the quality of seed, which can be evaluated by the seed testing ISTA 1996. The testing of seed quality is carried out on the seed sample drawn from seed lot to be used for cultivation. The quality of seed sample taken for testing in the laboratory is minute compared to that of the seed lot is represent. Method of seed testing, different type of method seed testing, seed sampling, physical purity, germination test, moisture. Seed sampling firstly seed sampling, the size and number of seed sample received in the laboratory is quite large. They need to be reduced to obtain working sample for carrying out various tests. That means the result can only show the quality of seed is taken for analysis. The seed size our such like if soybean sample we are received 500 gram seed sample, 500 gram seed for testing. Two lots are checking 100 two lots testing. Uh, it is important that the submitted sample representative lots from which they are drawn. The following info The following information will be mentioned in the label for sample which is to be checked and noted in the record that is maintained by laboratory: kind of seed, variety, number, date of the test, lab test number, seller name and address. This is a very important point. Physical purity testing. Next point, physical purity test tell us about the amount proportion of the pure seed present in the seed lot as well as the amount of other crop seed, weed seed, inert matter. If when we are testing of physical purity, lot of seed sample available, but what is the pure testing, physical purity testing? To identify First, to identify various species of the seed and inert particle comprising the sample. To know the percentage of consumption by weight of the sample that has to be tested. Uh, Third is germination testing. This test is very important in laboratory as well as field. If farmer, the purpose of laboratory testing of seed germination is to assess seed quality or viability and to predict performance of the seed and seedling in the field. A notified laboratory under seed X or a qualified laboratory of ISTA for seed testing must test seed proceed for sale. The ultimate aim to testing the germination in seed testing laboratory is to obtain information about the planting value of the seed sample by inference the quality of the seed lot. In addition, the laboratory germination result also required for comparing the performance potential for superiority of the different seed lots. Germination testing is very important, especially all crops such like vegetable, pulses, oil seed, germination testing of chickpea seed or germination on germination paper. Different type of seed lot we are checking in laboratory. Germination testing of chickpea. This is showing this picture, all of you. Germination chickpea seeds on specially germination paper checking. Next picture is germination testing of paddy seed on germination paper, showing of paddy seed, specially totally germinated. If root and shoot are specially grown properly, this is a normal seed. If root and shoot properly not growing in on germination paper, this is the abnormal seedling. Evaluation of germination test. The germination test evaluated as normal seedling, abnormal seedling, hard seed, dead seed. Normal seedling, seedling which has the capacity for continued development into the normal plant when grown in favorable condition. Soil, water, temperature, light. Normal seedling, seedling which has the capacity to continue development. The seedling development continuously in laboratory germination paper as well as in field. This is a normal seedling. Normal seedling root and shoot properly develop in on germination paper as well as in field. Abnormal seedling, seedling which do not show the capacity for continued development into normal plant when grown in favorable condition, soil, water, temperature, and light. This soil, water, temperature, and light, this is a very important because if water is not properly on seeds, this is not good for germination on seed. Next is hard seeds, such which do not absorb moisture till the end of the test period and remain hard. This is called as a hard seedling. Hard seedling do not absorb moisture or water. This is a hard seedling. So hard seedling, especially we are observed on especially pulses such like green gram, black gram, especially observed on hard seed. Do not absorb moisture or water till the end of the test period. Test period about 10 to 50 days, or after we can test till 1 month. And next point is dead seed. Seed which are neither hard nor germinated. Often dead seed collapse and milky paste come out when pressed at the end of test. This is called as dead seed. Dead seed is not germinated properly. Root, if sometime this is a root developed properly, but shoot is not developed properly. And next time shoot is developed properly, but root is not developed properly. This is a dead seed. Dead seed, we can observe various crops such like soybean crops, green grams, black grams, and some vegetable crops. And this is called as dead seed. Okra seedling stages. We can this is a picture of showing okra seedling stages. This is a picture adding in laboratory. Normal seedling. Normal seedling is the properly root and shoot. This is called as properly root and shoot growing. This is called as normal shielding. Other is abnormal shielding. Root and shoot are not properly developed. This is called as abnormal shielding. Last PPT we can observe on abnormal, normal, and dead shielding. And next is dead shielding. This is picture showing on okra seed. This is dead. Next, process of seed germination. Seed, firstly, we can sow in field seed and then develop some radicals and other some develop hypocotyledon root hairs. This is called as normal seed. Root and shoot are properly developed, and other fourth picture is properly develop whole plant. Root and shoot properly developed. Next picture is common garden piece. This is picture is total showing on normal crops. This is called as properly showing all root, shoot, stem, leaf. Some pictures hypocotyledon. Firstly, we can show seed and then germinate hypocotyledon, cotyledon. Two leaves, cotyledon, epicotyledon. Next slide is very important for especially in agriculture farmer. Soil temperature condition for vegetable seed germination in degrees Celsius. Different type of crops. This picture showing we are some observation on some picture just like cucumber. Cucumber is minimum temperature 15 degrees Celsius, the optimum temperature 21 degrees Celsius to 29 degrees Celsius, and maximum temperature 40 degrees Celsius. And day for germination 7 to 10 days. This is very important for soil temperature for seed germination testing. In laboratory, we can showing different type of observation in laboratory for such like especially vegetable crops, chili, tomato, brinjal, cucumber, onion. We can show 2 to 3 count. We can take a 2 to 3 count in laboratory. First count is near about 5 to 7 day. Second is 15 day. Third is 20 to 24. If first count is not showing properly grow in seed sample, we can take a second count. If second count is not showing properly root and shoot, we can take a third count. If first, second, and third count do not showing properly root and shoot, these crops such like cucumber, chili, tomato, and this is a dead seeds. Uh, we can showing cucumber 50 degrees Celsius minimum temperature, 50 degrees Celsius maximum optimum 21 to 29, maximum temperature 40, day to germination 7 to 10 days, and a minimum germination percentage. This column is very important. uh, Minimum germination is 80% for cucumber, broccoli 75% germination percentage, cauliflower 75 percentage, cabbage 75 percentage, carrot is low for 55 percentage, next broccoli 75 percentage, beetroot 65 percentage. Next is we can showing some importance crops. Onion. Onion is minimum temperature 0. Next, optimum temperature 21 degrees Celsius to 23 degrees Celsius. Maximum temperature 35 degrees Celsius. Day for germination in soil. Germination. If we can showing onion seed for in field. Defer germination in soil 10 to 14 days. In laboratory, we can take first count in 1 week. Next, minimum germination percentage of onion 70 percentage. Next, okra 70 percentage, peas 80 percentage, pepper 55 percentage, and etc. Moisture content of the seeds. Moisture is very important for seeds. And the seed moisture content is the amount of water in the seed. It is usually expressed as a percentage on weight basis in any seed testing laboratory. The seed moisture content is the most vital parameter which influences the seed quality and storage life of the seed. Seed moisture content is closely associated with several aspects of physiological seed quality. It is related, example, it is related to seed maturity, optimum harvest time, mechanical damage, insect damage. This is very important for seed mature. Sometimes we can showing seed germination, but if we can 100 seed sowing for in field, but some seed are mechanical damage, insect damage. This is not good for germination. Some showing of wheat field observation. Result of tomato hybrid purity testing on the basis of molecular laboratory. If last total PPT, we can take properly on quality control laboratory, and this PPT is especially on molecular biology laboratory. In molecular biology laboratory, we can showing properly 100% pure lot, basic, especially on vegetable called chili, tomato, brinjal, onion, as well as other pulses, oil seed, soybean, green gram, black gram. In molecular level, we can use in SSR marker. On that, this is called a DNA fingerprinting. DNA finger. This is a hybrid purity testing of tomato, showing banding pattern on molecular level. This is a DNA fingerprinting. Double banding means this is a 100% pure hybrid. Double banding means firstly this left side first band and second band. This is a female and male standard for hybrid purity testing. And second totally double band. This is a hybrid. This is called as hybrid. This is a 50% character goes to hybrid female and 50% character male. This is called as hybrid. And some showing one banding pattern. DNA ladder. This is a 100 base pair, 200 base pair, 300 base pair. Showing the what is the band showing on which base pair. This is called as 100 base pair, 200 base pair. This test is very important quality control lab as well as molecular biotechnology lab. In molecular biotechnology, we can show this is a picture of hybrid purity, and other picture we can show this is a parental purity. In molecular biology laboratory, we can different testing of molecular level, hybrid purity, parental purity, disease resistance, different type of spatially on 100% pure. This is called as well as we can differentiate data, field purity as well as laboratory purity, and this is called as what is the quality sheet. Next PPT is ELISA testing result for cotton leaf sample. So ELISA testing in India nowadays, BT testing, ELISA means BT testing, Bacillus thuringiensis, especially on cotton. Other crops are not, only cotton is the test for ELISA testing. Enzyme linked immuno linked sorbent assay. This is test is called ELISA testing of cotton. This is showing a yellow well. This is called as a positive sample totally. Cry1AC and Cry2AB genes added in this cotton, showing total of Cry1AC and Cry2AB. Thank you, all of you. Hello? Hello? Hello?
Yes, sir.
Hello?
Yes, sir. Thank you so much, sir, for providing a wonderful session. Now, I would request all the participants to unmute their mic and raise their question one by one, or you can also write your question in chat box. Participant, if you have any question, any query, you just unmute your mic and you can ask. Yeah, please ask. Yeah, please ask your question. Participant, if you have any question, any query regarding this topic, you just unmute your mic and you can ask. Sir, I think there is no question from participant side. You already resolved all the queries.
Okay. Is no problem.
Okay. So once again, I would like to thank Mr. B. Balerao, sir, for delivering a
Okay.
wonderful session on innovative lab to land technologies for Indian agriculture. Sir, your presentation provided us with deep insights. Many thanks for sharing your invaluable experience in agriculture technologies. Participant got benefit from your talk, sir. So at last, I would like to thank you for everyone for
Okay.
sparing their valuable time with us. Thank you so much,
Okay.
sir. Thank you, participant.
Thank you, sir. Specially thank to Dr. Kadam sir, Mayur sir. Specially give me a chance to present my PPT on this good webinar. Thank you so much, all of you.
Okay, thank you so much, sir. Participants, now I request you, please leave your meeting. You can leave your meet.